Hello students, welcome to 5-star classes. Today we are going to study chapter 2, Sexual Reproduction in Flowering Plants of class 12th. We will study this chapter into 3 parts. That is, first part would be about pre-fertilization events. Second part would be of pollination and fertilization. And the last part would be of post-fertilization events. In pre-fertilization event, we will study about the development of male and female reproductive organ, and later after development we will study about pollination and fertilization, that is fusion of male and female gametes, and later we will study about post-fertilization event, that is embryo development and seed formation. As we all know that flower is the reproductive unit of a plant. It has both male and female reproductive organ. It can be unisexual also or bisexual flower also, means it can only have male or female reproductive organ, or it can have both. See, now we will study androecium. Androecium, it represents the male reproductive organ. Gynoecium, it represents the female reproductive organ. Androecium is stamen. Gynoecium is pistil. Let's study about stamen more in details. Stamen, it has two parts. One is anther, another is filament. Okay, anther is a bilobed structure. Bilobed, it means it has two lobes. Each anther would have two lobes. And filament, it is a long structure. Now see in the diagram, it is the anther. The two lobes are visible here, and it is a long part, that is filament. Now see, if we have a transverse view of anther, it would be like this. This is the first lobe and it is the another lobe, and each lobe has two microsporangia. 1, 2, 1, 2. Each lobe has two microsporangia, so there are total 4 microsporangia. See, now microsporangia, they are surrounded by 4 wall layers. Microsporangia is surrounded by 4 wall layers. Let's see: epidermis, endothecium, middle layers, and tapetum. Epidermis and endothecium middle layers, they perform the function of protection and dehiscence. Later, the tapetum layer nourishes the developing pollen grains. Now see, in the center of this microsporangia, when the anther is just maturing, at that time there is a sporogenous tissue present in it. Sporogenous tissue is present at the center of this microsporangia. What will happen? Some cells of this sporogenous tissue, they would be microspore mother cell. Microspore mother cell, or you can say pollen mother cell. Some cells, they would undergo meiotic division. And we all know what is meiotic division. It is a reductional division and take place in two parts. This is the microspore mother cell. After meiosis 1, it would be divided into 2, and later after meiosis 2, it would be a tetrad, and this is known as microspore tetrad. From microspore mother cell, we are getting microspore tetrad through meiosis. So the process from PMC is pollen mother cell, development of pollen mother cell into microspore tetrad, or formation of microspore tetrad from PMC or pollen mother cell, is known as microsporogenesis. And all these tetrad or all these pollen grain, they are associated at this time, and later at maturity they dissociate and develop into pollen grains. And now pollen grain is the male gametophyte, means it will carry the male gamete. Now we will study the anatomy of pollen grain. Pollen grain is spherical and it is made up of 2 layers. Outer layer is exine, inner layer is intine. Exine, it is made up of sporopollenin, and sporopollenin is the most resistant organic substance. Due to presence of this sporopollenin, pollen grain can withstand high temperatures and strong acids and can be preserved for a long duration. Whereas intine is made up of cellulose and pectin. The outer layer at sign, it is not a continuous layer. It means certain apertures are present on it. See, these apertures are known as germ pore, and these germ pores plays an important role during fertilization. Now, inside the pollen grain, there are two cells. One is the vegetative cell, another is the generative cell. Vegetative cell is large and has abundant reserve of food materials, whereas generative cell is small and it floats in the cytoplasm of vegetative cell. It is spindle shaped. In over 60% of angiosperms, pollen grains are shed at two cell stage, and these two cells are generative cell and the vegetative cell. But in the 40%, it is shed at three cell stage, which means the generative cells divide by mitosis and form two male gametes. The generative cell divides by mitosis to form two male gametes, and now we have three cells. One is the vegetative cell and the rest two are male gametes. And now we will see some key points mentioned in the NCERT, that is pollen grain of rice and wheat loses viability within 30 minutes. And families like SRL stands for Solanaceae family, Rosaceae family, Leguminosae family, their pollen grain maintains viability for months. And now it is the end of this male reproductive organ, and now we'll start up with female reproductive organ in the next video. Thank you so much.